So in the previous lecture, we talked about the different macromolecules, as well as an introduction to molecular and cellular biophysics. One of the things that I had mentioned was that in order to build your way up to understanding how the different macromolecules work, we first have to understand the different forces of interaction within biomolecules. Once we understand how they interact and form macromolecules, then we'll be able to understand how they interact with other macromolecules, whether to create heteromacromolecules or in different processes. So first of all, we're going to start off with a recap. Then we're going to go through an introduction to molecular forces. Those molecular forces are then going to be broken down into intramolecular and intermolecular forces. So it'll be important for you to understand the differences between the two, which ones are stronger, which ones are weaker, and how we can model them using specific equations. So we won't be applying these equations until Physics 380 if you're interested in taking that, but in terms of just understanding the different forces, it's important to understand how strong they are and what they're proportional to. Afterwards, we're going to focus on hydrogen bonding because that'll play a key role in our discussions in future lectures related to different macromolecules. So let's go through a recap first of animal versus plant cell organelles, because that was the main gist of our previous lecture. Then we can take a look at the 4 macromolecules and some carbohydrates. So shared organelles in eukaryotes. These are the ones that are shared that are the most important. There is the nucleus, there is the endoplasmic reticulum, there's the Golgi complex, and there's the mitochondria. With the nucleus, the main thing is to remember it stores genetic information and it assembles ribosomal subunits. With the endoplasmic reticulum, know that it's attached to the nuclear envelope of your nucleus. The rough endoplasmic reticulum has ribosomes for protein synthesis, and the smooth endoplasmic reticulum has enzymes for lipid synthesis. The Golgi complex, basically an area inside your cytoplasm in your cell. It has stacks of flattened membranes called cisternae. The function is to process and modify the proteins that are synthesized. They also do post-processing. For example, it's going to create polysaccharides as well as create glycoproteins, a mixture of proteins and carbohydrates. And they also manage their storage and release. Out of all these, another very important one is the mitochondrion. It has a double membrane. The inner membrane has many folds. And its primary function is to provide power to your cell. So it goes through ATP production from enzymes catalyzing redox reactions. There were then unique organelles in animals versus plant cells. Lysosomes are primarily found in animal cells only because they contain hydrolytic enzymes and they break down ingested substances, cell macromolecules, and damaged organelles for recycling. Meanwhile, in plants, you don't really have them, but you have chloroplasts and large vacuole. So in chloroplasts, it produces ATP, very similar to mitochondria, but it goes through photosynthesis versus the electron transport chain. The structure of it is a double membrane, and inside your fluid or stroma, it contains thylakoids, which are stacked into grana. In plants, you also have this large vacuole. In green, I have this large blob here, essentially. It's a large membrane-bound vesicle that plays a key role in storage, digestion, and waste disposal. Apart from the organelles that we focused on, we also focused on just a general overview of the 4 great macromolecules: nucleic acids, lipids, proteins, and carbohydrates. Long story short, nucleic acids play a role in genetic information, the storing and transmitting of it. Lipids are going to play a key role in support and barrier formation, as well as the storage of long-term energy. Proteins are going to carry out functions within the cell, and carbohydrates are primarily used for energy and surface properties. In terms of carbohydrates, they provide energy to the body, specifically through glucose. There are also additional functions of carbohydrates that I think are very important. It also allows for energy storage. So your glucose can be stored as glycogen. And looking at this diagram here, the creation of glycogen, which is a polysaccharide, explains the process of essentially polymer formation quite well for a monosaccharide. So starting off from one glucose or one fructose, which is a monosaccharide, what happens is the two of these can go through dehydration synthesis and form a glycosidic linkage to form a disaccharide. The more glycosidic linkages that are formed and the more monosaccharides that are added on to the chain, it becomes a polysaccharide. And one such polysaccharide made up of a bunch of glucose subunits is glycogen, and it's got a bunch of branches to help with easy access to glucose whenever required. Next, you have the building of macromolecules. So not only do carbohydrates play a role in energy, they also can the monosaccharide itself can be converted into ribose and deoxyribose so that you can create your RNA and DNA. Apart from that, it's also very important for lipid metabolism. So through insulin regulation, insulin helps regulate your blood glucose levels. If you have too much blood glucose, then insulin kicks in to tell your body to use up some of your glucose so that you can lower your blood glucose level. But if there are problems with insulin regulation, then that means that you might end up with diabetic issues. So on to molecular forces. First, we're just going to go through a quick overview of molecular biophysics and why molecular forces play a key role in this. And then we're just going to take a look at why looking at molecular forces is important. Okay, so what is molecular biophysics? It's a subfield of biophysics that focuses on molecular structure and organization, the dynamic behavior of molecules at various levels of complexity. So what you can do is take a look at single molecule studies, or you can end up looking at supramolecular studies where a bunch of molecules are interacting with each other. In most cases, if you're taking a look at different nanotechnology tools that have been invented recently, like the atomic force microscope, those would be looking at single molecule studies so that you can pick out one molecule and study its interaction with another specific molecule, whether it be like a macromolecule or whether it be a system. So how do we start our study of molecular biophysics? And one of the key things before you start with any study is to look at its interactions. Being able to understand the structure of your molecule, the different charge or the different electrostatic properties of your molecule will allow you to determine its properties within the molecule itself, called an intramolecular force, or its properties with other molecules. So that would be an intermolecular force. So this is very important because it defines the structure and function of the molecule. And then once we have that, we'll be able to model it using different equations. So in terms of starting off, we first take a look at the chemical makeup of each macromolecule. So our goal is to be able to perform studies that will mimic these macromolecules in real life without its regular complexity. So with carbohydrates and lipids, we have hydrogen, carbon, oxygen. With proteins and nucleic acids, you have hydrogen, carbon, oxygen, nitrogen, and either sulfur or phosphate. So those are the chemical makeup of, those are the atoms that are included in those macromolecules, and they have different electronegativities or charges. So that means each of your macromolecules will have unique characteristics and electrostatic properties that play a role in its function and structure. So in order to understand the structure of biomolecules, just to repeat, we do have to understand what keeps them together, and that's going to be an intramolecular force. And we also want to understand how they interact with their surroundings, both near and far. So those would be intermolecular forces. As your macromolecules get assembled, so first of all, when we talked about carbohydrates, we started with a basic building block, which was the monosaccharide, then the disaccharide, then the polysaccharide. Now, when we take a look at the other macromolecules, their structure gets a little bit more complicated. Lipids have membranes because they are amphiphilic. They have both polar and nonpolar components. You have proteins, but they start off as amino acids and then they get combined together into proteins. And then you have your DNA starts with nucleotides and eventually they get stacked together to create your DNA helix, for example. So a macromolecule is going to require different interactions, both intramolecular and intermolecular forces, in order to create those structures. So your primary structure, which is usually a long chain of something, your primary structure is usually made of a strong force because you want to keep the core of your macromolecule stable. Afterwards, as it bends and it changes its shape or configuration, for example, a protein, maybe it's going to fold in a certain way. Those secondary, tertiary, and higher structures are then governed by weaker forces, so that they're going to be a little bit easier to unwind or denature. So how do we know if a molecule is stable? Energetically, what we can say is a molecule is a stable arrangement of two or more atoms. When it's stable, the energy of the combined system is going to be less than the sum total of your separate atoms. So I want you to understand why this is. It's because when you break the bonds between atoms, you need energy. This means that when atoms come together, energy has to be released. So that the individual energies must be greater than the combined energy because some is lost in the creation of that bond. So that would be very important to know, specifically for a short answer question. Now over here, it says that the energy in a strong bond is considered a few electron volts, while weaker forces are on a scale of approximately 0.1 electron volts. So now we can go into a little bit more detail about the differences between strong and weak forces, as well as what types of forces fall under which categories. Now let's take a look at the differences between intramolecular and intermolecular forces. Afterwards, we'll just focus on hydrogen bonding as a separate chapter of this lecture. So in terms of understanding the physical aspect of it, I would want you to know how to define and how to describe each of these forces. I'd also want you to know on a mathematical scale how strong these forces are. In terms of applying the equations, that is not something that I would focus on as much. So before we begin talking about molecular forces, we have to break a molecule down to its smallest component and then build up our theory of how these interactions can come to be. We start with the basic model of the atom, like this. And that's the building block of matter that can't be broken apart by any chemical means. So you have protons, which are positively charged. You have neutrons that are neutral, and they're inside the core or the nucleus. You then also have electrons that are going to rotate around that nucleus, and these electrons are negatively charged. You have these shells, so there's a K shell and an L shell. I won't worry about that now, but there are different orbitals and different levels of energy that these electrons can be at. So I'm going to first start with the Rutherford-Bohr model of the atom. Here I can see that I have rings or orbitals, and so each orbital seems to hold a different number of electrons. The n equals 1 orbital holds 2 electrons, the n equals 2 orbital holds 8, and so on and so forth. And there's a reason for that that we will explain later. As you get farther and farther away from the nucleus, there's an increasing energy of orbit for each of the electrons. So just remember those electrons are always going to try and fall towards your nucleus, because your nucleus is positively charged, whereas your electrons are negatively charged. So moving on, I have over here an equation, and it states that a photon is emitted with an energy of E equals hf. So I'll explain that in a bit, but the main thing is when an electron gets excited, or when it jumps a different energy level, it is going to emit energy because it has switched or changed its orbit. And so that energy is going to be proportional to Planck's constant times F. So in terms of the Bohr model, there are some things to understand when we draw them out or when we apply it. It's called the Rutherford-Bohr model because Rutherford first started out establishing that atoms consisted of a diffuse cloud of negatively charged electrons. So that's kind of like the first image that we saw. So then this negative cloud of energy or electrons was surrounding some positively charged nucleus. In 1913, Niels Bohr came forward and put forth the following 3 postulates about the Rutherford model, and so now it's just called the Bohr model for short, or the Rutherford-Bohr model. So number 1, based on the drawings we saw before, there were clear stationary orbits. So those stable stationary orbits are attained at discrete distances from the nucleus. And one of the postulates is that the electrons cannot orbit in between them. So going back, you can have an electron here, for example, you can have an electron here and here, but you can't have an electron just halfway between the 2 orbitals. So that is one of the postulates. The electrons have to exist within discrete states of energy of each orbital. Next, stationary orbits, where are they? So they're going to be at distances where the angular momentum of a revolving electron is an integer multiple of a reduced Planck constant, where n is the principal quantum number. So I'm going to talk about this a little bit more, but the key thing is that in order to satisfy this equation, your n can only be an integer number, that's 1, 2, 3, 4, and that's called the principal quantum number. And I'll draw a diagram to help explain why that is. Next, you have electrons that can gain or lose energy by jumping from one allowed orbital to another allowed orbital. And when it does so, it's going to absorb or emit electromagnetic radiation. And it's going to emit it with frequency V or F according to the Planck relation. So all you have to do is calculate the change in energy and then set it equal to H times V. And you'll be able to either calculate the energy or calculate the frequency of that radiation through your Planck relation. So this is an equation that you would have to know. Now you have the 4 quantum numbers. So I want to explain where these come from, and it primarily has to come from comes from spherical coordinates in 3D space. So the idea is that there are 4 quantum numbers that describe the position and the orientation of an electron in an atom. You have the principal quantum number n, which describes of the electron. Like before, I said it can only be an integer, 1, 2, 3, 4. The azimuthal quantum number, L, describes the subshell. So it gives the magnitude of the angular momentum through this relation. So you don't have to know this relation, but you do have to know that L can be 0, 1, 2, 3. So n starts at 1, L starts at 0. L is always going to be 1 less than n. So the value of L ranges from L equals 0, 1, 2 to n minus 1. We call L equals 0 the s orbital, 1 the p orbital, 2 the d orbital, and 3 the f orbital. So these are important ones to know. Next, you have the magnetic quantum number. It's going to describe the orbital or the cloud within a subshell, and it gives you the projection of your angular momentum along a specified axis. So it ranges from negative L to L. So I'll explain this in a bit as to what it actually means, because it has to do with the angle and direction that a specific orbital is pointing at. You then have spin quantum number. So that's represented by ms. It describes the intrinsic spin of the electron within each orbital, and that's usually going to yield either a spin up or a spin down as different states. So one of the things is we have these 4 quantum numbers, and just understanding what they are and what they describe is enough for this course. But in order to understand where they come from, we do have to talk a little bit about quantum mechanics. And this is something that if you're not familiar with, that's okay. So the Schrödinger equation, which most of you have probably heard of, is considered a partial differential equation that describes the quantum state of some physical system. When we take a look at the atom and at electrons, we only concern ourselves with the wave function. So another way to read Schrödinger's equation is that the sum of the wave functions, kinetic energy and potential energy, is equal to the total energy of the wave function. So that psi, the thing that looks like a y over here, that represents your wave function. And so here you have the kinetic energy, here you have your potential energy, and here you have your total energy. So it's this is called a partial differential equation. Now, the most important part is that from the wave function, the Schrödinger equation delivers our 3 quantum numbers, and each combination can have spin up or spin down. So our goal is to just figure out where those numbers came from, the quantum numbers. So if we focus on the wave function psi, it has to exist in 3D. It means that its solution must also have 3 parts. So your wave function must be defined by some sort of radius, some sort of angle, and another angle. And then each of these can have a value. The solution will be some combination of these 3 values to give you a specific position in 3D space that can be spin up or spin down. So drawing this, you can do a quick sketch in terms of what this means. I would have, let's see, I even have space for this. I can have my X, my Y, and my Z axis in 3D space. And in green, I can, let's just create some sort of sphere here. And maybe I want to take a look at this position right here on the surface of that sphere. That's the exact location in 3D space that I want to look at. That specific location is a certain distance r from your origin. And so that's why your r represents n. And that would be the distance away from your nucleus or your origin at discrete integer values. Next, you have, in order to get here, even though I have a specific radius or a distance from my nucleus, I still need to know where it is in terms of the xy plane and the yz plane. So I have theta, and theta is usually the angle away from the z-axis, and that's going to be considered the azimuthal angle or the angular momentum angle. So you have that. But even though I have this angle from my z-axis for my theta, I can still spin this around 360 degrees. I don't know exactly where to stop, so I still need a further measurement. And so this angular momentum is usually considered L. This one is usually considered N. And then I'll put it in blue. I have a rotation specifically around here. Let's just let's do that. That would be our phi. So that's going to be the angle on the xy plane that your specific electron is going to be at. And this is going to be considered your final value, your ML, magnetic quantum number. And so at this spot, over here, you end up having either spin up or spin down positions. So that is exactly how I like to envision the magnetic or your different quantum numbers. Of course, there's a lot more consideration to be placed into each, but if you are to try and understand where they come from, it's because of the wave function and because it's defined in 3D space that we have to give each of the solutions a specific value, and that's where those numbers come from. Okay, so next, now that we kind of know that the solution to a wave function can help us derive the quantum numbers, we have to talk about the Pauli exclusion principle. So the Pauli exclusion principle states that no two electrons can have the same 4 quantum numbers. So that means in one position, like I drew before, you can have the same position but spin up and spin down. So there's 2 options for each location. And so since an orbital can contain a maximum of 2 electrons, each is going to have an opposing spin. So one has to be plus or minus 1/2. So the 1s, I'm just going to go through an example. Argon, which has an electron configuration representation of 1s squared, 2s squared, 2p6, 3s squared, and 3p to the power of 6. So key thing is argon, if I look at the periodic table, it has atomic number 18. It means it has 18 electrons. The number in the subscript over here represents the number of electrons. In that level. And of course, they are ordered in terms of increasing energy. So the 1s level, if I take a look at this, this is a typical electron configuration diagram on the left. The 1s level can accommodate 2 electrons with identical N, L, and ML quantum numbers. So that means for each of these, there can be spin up and spin down. The 2s level electrons have a different principal quantum number to those in the 1s orbital. So now you have n equals 2. But the pair of 2s electrons are going to differ because they have opposite spins. So you still have 2 over there. Then you have 2p because they have a different L value. And in this case, that means they have a different orbital angular momentum. So we use P instead of S. And because you have 3 p orbitals of energy, it means they can exist in the x, y, and z plane, which is why I drew that spherical coordinate system so that we can determine where they are as right angles to one another. So a similar pattern is can be mimicked for the 3s and 3p orbitals as well. So in terms of understanding this, I would just know what the Pauli exclusion principle is and whether you can have the same 3 quantum numbers, same 4 quantum numbers, and why. So moving on, let's take a look at the ionic bond. The ionic bond is formed when electrons are transferred from one atom to another. One example is sodium chloride. So sodium usually has one electron in the valence shell. And so we'll just assume this is sodium, and that this exterior circle is your valence shell, the outermost shell of your electron cloud. So it has one electron, whereas Cl has 7 electrons. What happens in an ionic bond is that an electron is transferred from one atom to another. So in this case, Na would donate 1 electron to chlorine, and both of them become charged. Na loses an electron, becomes positively charged. Cl gains an electron, becomes negatively charged. So then, because one is now positive and one is now negative, they experience electrostatic attraction to form an ionic bond. And this sort of bond is the strongest bond there is, because there is no temporary or momentary change in electrostatics. It's almost considered permanent. So moving on, we're going to a weaker type of bond called a covalent bond. It's formed when electrons are shared between 2 or more atoms. So a molecule is a cluster of atoms held together by covalent bonds. So covalent bonds are going to be able to create molecules. So once again, I have the same 2 atoms. In the valence shell, I have one extra electron, and here I have 1 extra electron. But maybe the 2 of them require just 1 extra electron or to satisfy their orbital. So that means if they share and they satisfy their valence shells to become more inert, then a molecule is created through a covalent bond. And so that bond length between the 2 nuclei, between the 2 plus signs here, is going to be determined by how much attraction and repulsion there is. So how many covalent bonds can there be per molecule? So one of the things to note is that there can be more than 1 covalent bond per molecule, and how can we determine how many there can be? So in terms of the atomic number and the number of electrons in each atom, we take a look just at the outermost shell, the valence shell. So over here, for hydrogen, there's 1 electron. It's not complete. It requires 2 to complete its first shell. In the second shell, you need 8 to complete the second shell, and another 8 in the third shell for a complete valence shell. So the important thing is that if there is an uneven number, or if there is an imbalance or incomplete valence shell, the atom will want to either get more or get rid of its electron so that it can become more inert. So taking a look at carbon here, it has 4 and it's missing 4. It means that it can bond with one other type of atom that also has 4 missing electrons. So that would create one bond, or it can bond with 2 other molecules with that just require 2 electrons. In that case, it forms 2 double bonds. Or it can form 4 single bonds with, for example, hydrogen. So you get CH4. So that hydrogen kind of shares 1 electron with carbon, and another hydrogen shares 1, and so on and so forth, until carbon is sharing the electron of 4 hydrogens to fill up its required 8 in its electron shell, and hydrogen shares 1 electron with carbon so that it fulfills its own electron shell. So how many covalent bonds per molecule will really depend on which 2 atoms you're trying to combine with each other. Okay, so then I want to take a look at the electronegativity of intramolecular forces. Covalent bonds and ionic bonds are both considered very strong bonds, and we can differentiate them from their bond polarity. So the more polar covalent bond gives it greater The more polar a covalent bond, the more it becomes an ionic bond. So a covalent bond will never be an ionic bond, but the more polar the covalent bonding that occurs, the more ionic character it has. So the stronger it gets. So moving on to intermolecular forces and weak interactions. Remember, the two strong interactions are ionic and covalent bonds. We now want to take a look at weaker interactions. So these are considered long-range weak forces between molecules, and they are primarily concerned with defining secondary and higher structures in macromolecules. So once the primary structure of your macromolecule has been created through specific bonds and linkages, then what we take a look at is how they fold and how they interact with each other to create different configurations of your macromolecule. So different configurations and different shapes of those macromolecules will affect their function as well. So the main intermolecular forces we want to look at are the electrostatic force, charge and dipole interactions, van der Waals forces, and the three types. And then we're going to take a look at hydrogen bonds in the last section. So we'll go through these fairly quickly, but the key thing is just to note what the force and the potential energy is proportional to. And we just want to be looking at the distance between the two ions or dipoles or charges. So the first and the strongest weak force that we're looking at is called the Coulomb force, or the electrostatic force. It's a force between charged particles, so really we're looking at two ions. And so we can describe each ion as just a simple point particle and apply Coulomb's law of electrostatics. So the interaction between 2-point charges, little q and large q, are separated by a distance r in a medium of dielectric constant epsilon r. The force between two charges, so one charge to one charge, is going to be QQ over 4 pi epsilon naught, and then epsilon r, which is 80 for water, times r squared. Keep in mind, 4 pi epsilon naught is also 1 over 4 pi epsilon naught is equal to K, Coulomb's constant. Next, you have a charged dipole force. So maybe you have a charge, and then maybe your molecule is actually non-polar and is polar and has a dipole. So in that case, you take a look at a point particle, and unfortunately, you now have to introduce something called an electric dipole moment. So the calculation gets a little bit harder. Your dipole moment is going to be the charge of your dipole multiplied by its direction or its orientation, and here it's kind of at an angle. So its contribution to a specific potential energy that you're trying to measure will change depending on how it's oriented. So in general, your potential energy is going to be proportional to 1 over r cubed. And that's all we really want you to know. Over here we have U is proportional to r cubed, 1 over r cubed. Now with van der Waals forces, it applies to all of your weak attractive forces. So generally, these are considered momentary attractions between molecules and individual atoms. They're considered unstable, and they're primarily momentary polarized particles or induced dipoles. But these van der Waals forces are very important. They're behind phase transitions and as well as bulk properties. So some examples of phase transitions would be the freezing of liquids to solids, condensation, as well as with bulk properties, you have viscosity and hydrogen bonding. So what types of van der Waals forces are there? There's 3 types. Hydrogen bonding is separate. There's the Keesom force, when you have 2 dipoles interacting with each other. You have the Debye or polarization force, which is from any non-polar molecules, but they can get induced dipoles. Then you have London dispersion forces, which are between 2 sets of, or 2 induced dipoles. So all of these weak forces are usually at or smaller than 1 over r to the 7. So with a Keesom force, you have a dipole-dipole interaction. And so I've kind of represented this as two different dipoles, and they have two different orientations. So if you want to do some vector calculations, this gets fairly complicated. But the most important thing is when the two dipoles are lined up horizontally, then you have an average potential energy of that's proportional to 1 over r to the 6. So that's important. So just remember, Keesom forces will be proportional to 1 over r to the 6. With the Debye force, you have induced dipoles. So you now have a charge, and then you have a nonpolar object that has an induced dipole because the charge induces some sort of temporary charge on your nonpolar molecule. Now, I won't go through this description in detail, but essentially we also have your potential energy being proportional to 1 over r to the 6. One key thing to note between this and the Keesom force is that the energy in the Keesom force is proportional to temperature, whereas this one is not. Now, the last one is called a London dispersion force, and that's the weakest intermolecular force there is. It's between two induced dipoles. They're present between all molecules, and they're very fleeting or temporary. So it occurs with both polar and nonpolar molecules, and the range is approximately 0.2 to 10 nanometers. Now, the potential energy is also proportional to 1 over r to the 6, but it is proportional to the frequency of quantum oscillators in its ground state. So these have to do with different quantum fluctuations within each of these molecules. Now onto hydrogen bonding. So with hydrogen bonding, I just first want to go through what it is, take a look at how they play a role in water and macromolecules, and then go through some hydrophilic and hydrophobic forces. So we all know what water looks like from the previous lecture. Hydrogen bonding, or a hydrogen bond or an H bond, is an electrostatic force of attraction between, number one, a hydrogen atom that's covalently bound to a more electronegative group, like O, N, or F, and two, another electronegative atom bearing a lone pair of electrons. So hydrogen bonds can occur intramolecularly or intermolecularly. So when I take a look at water, I care about water more because all the macromolecules exist in water. So the water molecule has a net neutral charge, but because it has an electropositive region and an electronegative region, it becomes some sort of a dipole or a polar molecule. So what happens is if you have a bunch of water molecules together, you create a hydrogen bonded network, and they're cohesive, which allows for surface tension and other unique properties. Now, where do hydrogen bonds exist in macromolecules? They can exist in proteins. We'll go through this in a lot of detail in the next couple of lectures. But here, if you have a protein and you have a bunch of amino acids that are chained up together, you have an option to either fold the protein into an alpha helix or a beta sheet. And that decision comes from the types of hydrogen bonds that occur between different groups. So for an alpha helix, hydrogen bonds have to occur between the NH and the CO groups of neighboring peptides within one strand. For a beta sheet, your hydrogen bonds are going to be between peptide bonds in different strands, so you end up forming a sheet instead of having a twist. Hydrogen bonds in DNA are very important as well. We have hydrogen bonds between your different DNA base pairs, which we'll go through in detail as well in a future lecture. But there are 2 hydrogen bonds between thymine and adenine, and there are 3 hydrogen bonds between cytosine and guanine to create our AT and CG base pairs. Last, you have, I just want to briefly talk about hydrophobic versus hydrophilic forces. Water is going to force hydrophobic groups together, and hydrophobic means water fearing. So if you have some hydrophobic substances in water, they're going to try and group together to form a hydrophobic core or a hydrophobic group. Hydrophilic molecules will want to be exposed to water. So if you have a hydrophobic core, or if you have a hydrophobic molecule with some hydrophilic or polar components to it, then those ones will most likely be presented on the exterior surface to interact with water. Now, when we talk about lipids, lipids are primarily amphiphilic, and that means they have both hydrophobic and hydrophilic parts. And because of that, it drives the self-assembly of lipids because your polar molecules will want to orient themselves at the water surface, and there will usually be a hydrophobic core where no water can get in. So as a summary, what should you focus on? We should focus on the importance of molecular biophysics. We should figure out what types of molecular forces there are. So just understand what the potential energy of different intermolecular forces are dependent upon, whether it's temperature, quantum oscillations, and just understand what types of ions or charges or dipoles we're looking at for each type of intermolecular force. Know the difference between ionic and covalent bonding, and of course, have a brief understanding of hydrogen bonding because we'll be taking a look at that in different macromolecules for different future lectures.